Hello everyone. Today we're going to be presenting the study conducted by UJ Kao et al, which was done in very recent years, and it's called "Switchable CAR T cells outperformed traditional antibody redirected therapeutics targeting breast cancers." So we're going to be focusing on the switchable CAR T cells, which are modified T cells from the immune system, which will help fight the cancers in general, but especially breast cancer. So it's a very novel method that that is currently being incorporated, and we will see what are the benefits of this method. So first, we're going to start with the introduction, of course, then the methods. We're going to be a little bit fast on the methods because it's mostly chemical reactions, which we don't, it's not extremely important to know the details of them. It's not reflected in the results. Then in the results, we have 4 parts. The first is how did we design the CAR T cells? How did we design every molecule that's going to be used? Then there will be an in vitro and an in vivo cytotoxicity assay. And finally, the novel bispecific sCAR T cell strategy is going to be studied. We mean by this that the first 3 parts are going to be like a general study of the cytotoxicity, and the final will talk about why the bispecific sCAR T cell strategy has many advantages. So let's start by an introduction. So the big question that we always start with is, what is immunotherapy? So we all know that it's a type of cancer treatment that uses the substances that are made by the body or in a laboratory to boost the immune system and help the body find and destroy cancer cells. So currently, there are 3 antibody-based therapies that are the most used. We have ADCs, we have bispecific antibodies, and CAR T cells, which are also called chimeric antigen receptor T cells. We're going to be focusing on CAR T cells, but in the whole presentation, we're going to compare to the ADCs, which are antibody drug conjugates, and we're going to compare it to the BSABS. So, okay. Previous papers were done on BSAB, and it showed that BSAB was superior to ADC when concerning the breast tumor that has low antigen expression. And I'm going to focus here because we're going to be carrying this idea. We have to have this idea in mind the whole presentation. The biggest advantage that we can get from the CAR T cell therapy that we're going to study is the fact that this CAR T cell therapy works on breast cancers, on the cancer cells that produce low antigen expression. The immune system, when there are antigen expressions that are too low, sometimes it cannot attack, it cannot protect the body because the antigen expression is too low. But using the CAR T cells, we're going to see later that even with a small production of, in this case, HER2 receptors, we are able to fight the cancer cells. So as a general explanation, CAR T cell, basically we take the T cells from the blood of the patient, then there's a reprogrammation which will modify the T cells into CAR T cells. Of course, multiplication is done, and the blood is infused into the patient back, and those modified CAR T cells will attack the cancer cells. So the aim of the study is, as we said before, to compare the ADC, BSAB, and CAR T cells therapies, but also to explore the bispecific sCAR T cell therapy. So the materials and methods. All the breast cancer cells here were obtained from the American type culture collection, and the all cell lines were at very specific parameters that were predetermined to enhance the growth and to have a very basic parameter that could be in the body. So what's FITC linker? FITC linker, it's gonna be used for the recognition of what's happening. It's for the fluorescence. So here we have the name of the molecules. We're not gonna dig into it too much. It's not our main point of interest, but it is still important to mention. Now, how did anti-HER2 FITC was prepared? First, the anti-HER2, which is the antibody, obviously, was linked with FITC, which is a fluorophore, which will show fluorescence during the experiment. This was done through a site-specific conjugation method. This conjugation was achieved through a series of chemical reactions that we'll also not dig into because they are very they are not in the scope of the study, but they were done to completely get the anti-HER2 FITC. So why did we choose FITC? It's because it will let us label the anti-HER2 Fab for further use. That means that by using the fluorescence, we're not doing anything invasive. The fluorescence will not impact the cells or change it and make it unusable later. We chose the fluorescence because they will be reusable later on. Now, for the preparation of the bispecific antibodies and the ADC. The bispecific antibodies in this case, it's anti-HER2 Fab with anti-CD3 Fab. And their preparation was done using genetically encoded unnatural amino acids using orthogonal chemical reactivity. While for the ADC, by using site-specific conjugation, we linked anti-HER2 IgG to something that we call auristatin. This was done using the PAC-P, the PAC-PHE incorporation, and something that we call oxime ligation. So to transmit the cancer cells, we needed a vector, and the vector used lentivirus. So many laboratory procedures were done for the production of the lentiviral vectors, and they were used in transducing the human T cells, the human CAR T cells, basically. And so to know if the surface expression of CARs was done, to know if the reaction was done, we used a common technique, which is flow cytometry. Now, to check the binding of the antigen-specific switches to the target cells, we used also cytometry, which allows for the quantitative analysis of the cellular properties, like the surface receptor binding in this case. So flow cytometry was used a lot. Let it be for the lentivirus vector part, where we saw if the T cells, if the CAR T cell modification was done properly, and also for the binding of the antigen. For the in vitro cytotoxic assay, basically the CAR T cells were put with the target cells in a co-culture. It's a very, very common way to do the cytotoxicity assay. And we measured to see the cytotoxicity, to see if the cell was actually functioning to kill the other cells. LDH release was measured, and the results were expressed as a percentage of the cytotoxicity. So once again, flow cytometry was used here to measure the activation of the CAR T cells. And another thing that was measured was the levels of the key cytokines, which are produced. So the CAR T cells that produce cytokines, which will help in the immune system fighting against the cancer cells. So the levels of the key cytokines that were produced by the CAR T cells were also measured to see the efficiency of the CAR T cells. For the in vivo, 8-week-old female, of course female because it's breast cancer, and NSG mice were taken, and there were this is very important. There are 4 types of tumor models, 3 types of tumor models, and one that's not mentioned. It's not a type of tumor model. The last one, it's like a control. So here we can see that we took HER2 3+, HER2 2+, and HER2 1+ cells. So what are the pluses? 3 plus, 2 plus, 1 plus. HER2 1 plus produces the least of the HER2 antigen in this case. So we're going to see later that it's different different types of cancer cell types. It's different cancer cell types, but the plus next to the HER2 shows how much HER2 will be produced by these cancer cells, and obviously the more HER2 is produced, the easiest the immune response will be, because it will detect that there's a lot of HER2. The anti-HER2 will detect a lot of the substance, and they will come to attack it. So finally, for the bioluminescence imaging, as we said, BLI is very useful, and it was chosen because it is non-invasive, and we'll be able to use the subjects again later. Now on to the results. The precision designed HER2 targeted immunotherapy. So this is basically how did we do the how did we construct the antigens, the antibodies that will be used. So from PACF, we're able to produce the ADC, the BICB, and the sCAR T cells. For the ADC preparation, by linking an anti-HER2 IgG with auristatin F linker, we obtain the ADC, which will be used later to show to compare with the sCAR T cells efficiency. So at left, we can see the bispecific antibody, how it was produced. Basically, as we said before, it's by linking anti-HER2 and anti-CD3. We did the bispecific antibody, while at right, we showed how we linked anti-HER2 with FITC for the fluorescence. I'm not digging too much into the explanations here, because it's not directly correlated with the results that we're going to get. This is a conventional way of producing the antibodies. So here, this is just to show that the fact that the reactions produced the right amount of, for example, the bispecific antibodies, which we can see first. We had more than 95% of conjugation products, and this was determined at left by mass spectrometry, and at right by using polyacrylamide gel electrophoresis, also known as the SPAGE. So here it's what I was talking about before. We can see that HER2 3+ will produce, I'm going to try to zoom in, 10 to the power 4 fluorescence. We saw 10 to the power 4 of FITC expression, which shows how much HER2 is produced from the HER2 3+. While for the HER2 0, which is like a control, there was no fluorescence that was produced outside the normal fluorescence levels. This is important to see if the cells, for example, the sCAR T cells, nobody tells us that they won't be harmful to normal cells. Normal cells in this case are represented by the HER2 0, which do not produce very different amount of HER2, and it's like a safety check. If the sCAR T cells also attack the HER2 0, it means that it can attack not only cancer cells, but also cells around the body, which is obviously what we don't want. So, and this is shown here that for ADC, for BSAB, and for anti-HER2 FITC, the three of them do not attack the HER2 0 because they are the control. This is shown by the, they're not expressed for the HER2 0. We don't know yet about the cytotoxicity. We can see there is 0 level of fluorescence intensity, while for the SKBR3, which is HER2 3+, which produces the most HER2, we can see a strong fluorescence intensity for every case except the control, of course. Now on to the quantitative results of the cytotoxicity between ADC, BSAB, and CAR T cells. So here we have many cell lines, many cancer cell lines, but I want you to focus the most on HER2 3+, on HCC 1954, and on MDA MB468, which is on the bottom right. So we can see that on the HER3+, the triangle, which is sCAR T cells, had a way higher cytotoxicity percentage than the other ones, than ADC and BSAB. This is applicable to all of the results, as we can see here. I was going to say to most, but not to all of the results. And also, it's very, very important to focus on MDA MB435. Look at HER1+. As we said before, HER1+ will not produce a lot of antigen, will not produce a lot of HER2 antigen. But ADC, for example, couldn't even recognize the HER2 because of the absence, because of the lower number of HER2 antigens, while sCAR T cells and BSAB recognized it, but of course sCAR T cells had a higher cytotoxicity than BSAB. Oh, this is just the numerical values of what we presented before. We're not going to dig into it. The graphs show enough already. So this shows the results that, like, after the scientists, they saw what the sCAR T cells do, they tried to experiment doing, placing CAR T cells with conventional anti-HER2 antibodies. And as we can see, most of the results were very conclusive, except of course for the HER2-0, which also enhances the belief that it won't affect the normal cells. We can see 75% of cytotoxicity for all of the results. Some maybe for HER1+, for example, because it releases less HER2, the results were around 60%, approximately 60%. But for all the results, there was cytotoxicity observed that was way more than 50%. Now onto the in vivo anti-tumor efficacies of the immunotherapeutics. It's going to be very similar to the previous results because as we can see here, for the HER2 the time that was compared is very important. It shows that even after 72 hours after the CAR T cells were infused, there was still cytotoxicity observed, and it didn't decrease. Even after 48 hours, it was at its peak, which shows that it can be a viable, a usable, a practical method. For example, if someone has cancer and want to do immunotherapy, if it showed that every 3 days CAR T cells were still that cytotoxic, it would show that, for example, this is very far-fetched, but the person that goes to the hospital wouldn't have to go every day and infuse new CAR T cells if they are efficient on the long term, on the long run. So this is something that must be studied again in further studies. But anyway, here we can see that the tumor volume, of course, I'm speaking here at the top right. At the top right, the tumor volume kept increasing for the control because obviously we didn't put any CAR T cells. But, and this is very important, for the sCAR T cells, it went down to 0 millimeters cube. We can conclude that the tumor was dead at that point, but because the study doesn't go deeper into, for example, it doesn't do other methods to recognize the tumor cells, we can't really advance on this. So here also the tumor volume was shown to be the lowest for sCAR T cells and for the control sCAR T cells. Then you have the BSAB, which was, as we said before, BSAB was shown to be more effective than ADC, but it's still, the tumor volume was still high. It was still 600 millimeters square. And this is important too, because ADC showed not no result, but a very, very, very small result, represented by the high tumor volume, around 1,500 millimeters square. 2 seconds. Oh. Okay. Here it's also the same way of thinking. It shows how sCAR T cells also for the luminescence, they show the zero result, which means that the tumor size was so small that we couldn't observe luminescence. So here at right, we see that the sCAR T cells showed better results than the ADC and the BSAB. If is it zooming in your screen or only in mine?
No, it's working perfectly.
Okay, we can see that the sCAR T cells, which are the triangles upside down, showed a 24 millimeter square tumor volume, 24.5. While for the BSAB, for example, it showed a 28. So in all the results, the bispecific CAR T cells, the specific CAR T cells were more efficient than the BSAB and the ADC. So now the final results about the novel bispecific sCAR T cell strategy. So here we use two receptors, HER2 as showed before, HER3 plus produces the most, HER0 doesn't produce a lot, and IGF-1R, which is present in many cells, and it was used to see if the cell will be cytotoxic against itself. So IGF-1R, it's not specific to cancer cells. It's produced in many different cells. And as we said before, the most important thing is that the treatment won't cause autoimmune diseases. So this is a quantitative result of what was shown before. Shows that anti-1GF-1R had a normal, it was a normalized quantity, while between the HER2 3 plus and the HER2 2 plus, we obviously have more HER2 in the 3 plus, and it goes down until HER0. Here is, I think, the most important result of the whole study. This is a personal opinion, but I highly believe so. For HER1 plus, the only CAR T cell, the only result that was observed for HER1 plus, which produces low levels of HER2 antigens, was for the bispecific CAR T cell. Now, what does this mean? It means that by using bispecific CAR T cells, we are able to target even minimal levels of HER2 antigens, which is a mechanism that many tumor cells use. They don't use it, but many tumor cells survive the immune system because of their low antigen concentration and quantity. So by using the bispecific, which can attack even the lowest concentrations of HER2 antigens, we can we can add efficiency to this treatment by attacking even the smallest amounts of HER2 antigen. Those are also numerical values. We can see that the IC50, which is the immune inhibitory concentration, so it's the concentration needed to inhibit the tumor growth by 50%. It was obviously the lowest in bispecific CAR T cells, and many results were non-defined for the sCAR T IGF-1R, for example. So as we said before, the IGF-1R, it's very important to note that there was no result for all the cancer for all the cells that were studied, which is positive, because if we had the results, it would mean that it could attack any cell of the body and cause autoimmune diseases, and it would be counterproductive. So to sum up this this paper, a switch redirected CAR T cell strategy achieved superior performance to BSAB and ADC. This was in all results, as we showed, for the eradication of cancers with low antigen expression, and allowed for safer application to overcome toxicity related to persistent conventional CAR T cell therapy. Also, the versatility of the multi-redirected sCAR T strategy contributed to addressing the overall antigenic landscape of cancers. So what we say about the overall antigenic landscape of cancers, it basically talks about the fact that antigens, let them be at a low or at a high concentration, will be attacked, and cytotoxicity will be observed.